Isolation of lactic acid bacteria from commercial and homemade yogurt. Our group have 9 people, consists of 4 boys and 5 girls.
For the objective, the first objective is to isolate the LIB potentials in store-bought and homemade yogurt by using MICROBIAL. For the second, to identify and compare the characteristic and assess the LIB that presents in commercial and homemade yogurt. The study's goal is to determine and characterize the antimicrobial activity of lactobacillus producing bacteriocin isolated from curd samples. Lactic acid bacteria are a type of industrial microorganism that is used in the production of fermented foods as dietary supplements, probiotics, and even cosmetic ingredients. Preparation of homemade yogurt. Okay, first we take 500 ml of cow milk and then we heat the milk to 80 degrees Celsius. Put 5 tablespoons of yogurt, which is Lactic brand, as the starter to culture the LIB while cooling them. And stir the yogurt starter with the rest of milk and incubate it for 7 to 9 hours. For commercial sample, we use Just Lady Yogurt brand. The brand is important to highlight as the brand may use either 2 types of bacterial strain to produce lactic acid based yogurt, Lactobacillus sp or Streptococcus sp.
As the result, the texture of the yogurt is soft, silky, and pudding-like, while the taste of the plain yogurt has a light, airy taste and tangy. And last, the odor of the plain yogurt has a mild and almost inconspicuous of aroma that is agreeable to the nose. Preparation of medium odor of MRS.
Preparation of MRS saga. First, we take 33.35 grams of MRS and then suspended it to 500 ml of distilled water and heat it by doing sterilize by autoclaving it at 121 Celsius for 50 minutes and then cool it to 50 degrees Celsius, mix well and pour it into sterile petri dish.
Dilution of yogurt sample by using commercial and homemade yogurt.
Dilution of yogurt sample. A sterile pipette is taken. 1 ml or 1000 microliter of properly mixed sample is drawn into the pipette. The sample is then added to the first tube to make the total volume of 10 ml. This provides an initial dilution of 10-1. This step is repeated until it reaches the 6th tube, which it make an initial dilution of 10-6.
Next, for the spread method, firstly, sterilize hand and table using ethanol with a wiping cloth before performing lab conduct. Then, by using a micropipette, take 0.1 ml of final dilution and put it onto the center of the surface of an agar plate and dip the L-shaped glass spreader into alcohol. Next, flame the glass spreader, which is hockey stick, over a Bunsen burner. After that, spread the sample evenly over the surface of the agar using the sterilized glass spreader and incubate the plate at 37 Celsius for 24 hours.
As the result for the homemade samples, the colonies become decreased, and the final result show that the colonies below than 2000. For the commercial samples, the colony also become decreased, and as you can see on CD dilution C, show a clearly single colony with 206.
Next, let's conduct the split method. Firstly, sterilize and wipe the surface that will be used to perform the split plate method. Flame the loops until hot and leave the loops for a while. Take the bacterial colony using a loop that has been sterilized from the bacterial colony plate and streak the bacterial colony above the MRS agar. The streak plate method is important for the growth of single bacteria to live well and purify the strain free of contaminants. Streak plate method is done so that single colony bacteria can live with perfect pure genetics and facilitate the task of counting colony bacteria. As a result, you can see how the streak for homemade samples and streaks for commercial samples were produced.
Biochemical test of grain staining.
Let's come to the grain staining process. First of all, we need Bunsen burner, a loop, a slide, and grain staining reagents. First, stir up water was placed on slide by using the loop. Second, the suspended culture transferred from MRS agar to the slide by using the stirred loop. Third, the slide needed to dry through Bunsen burner. After that, crystal violet added a few drops to stand over the fixed culture for 1 minute and rise with distilled water until the colors wash off. Next, iodine solutions added to cover the smear for 1 minute and also rise with distilled water. The step was repeated with acetone, which is decolorized, and also safranin for 30 seconds, 1 minute respectively. When the slide dries, examine the slide under the microscope. According to the research of Moyana, say that LAB is one of the groups of gram-positive bacteria with non-spore forming cocci or broadshed. Now, let's see the result under the microscope. For homemade samples show the significant result of gram-positive bacteria with cocci shape, while commercial sample show the significant result of gram-positive bacteria with rod shape.
Catalase test.
Next is a catalyst test. First, smear the culture sample on the slide, then add a few drops of hydrogen peroxide solutions on the slide and observe for immediate bubbling. As a result, lactic acid bacteria produce oxygen by forming bubble of gas. According to Ismail, lactic acid bacteria do not produce catalyzed enzymes that converts hydrogen peroxide into water and oxygens and does not indicate the forming of bubble gas. However, in the journals of food science, some lactic acid bacteria species can form catalyst activity, which this activity known as fusel catalyst activities, and can be found with species of lactobacillus bacterium in yogurts by depending on the substrate used as an energy sources. Additionally, the test might be get a false negative on positive reactions based on the England said that.
Oxidase test.
After that, the oxidase test is occurred. The tetramethane p-phenyllithium diamine dihydrochloride was added few drops on the filter papers. Then the colony of bacteria is added and gently smeared on the filter papers and observe the immediate purple colors. As a result, it indicates with no color changes after adding a few drops of reagent to the filter papers means that the LAB are negative results. However, there are some error could happen. The reaction usually after 30 seconds oxygen surrounding with effect the result of color changings, and also the reagent must be used not more than 1 week. Additionally, the nickel steel and other wire loops can give false positive result. Thus, recommended to use platinums or inner transfer loops such as sterile wood stick, toothpicks, and sword.
As the conclusion, LAB colonies can be reproduced using MRS agar, which has suitable temperature and pH agar characteristic. Anaerobic bacteria thrive in an environment rich in MRS, which contains carbon, nitrogen, and vitamins to aid their growth. In addition, the biochemical test of the gram staining method may distinguish between characteristics of LAB in handmade and commercial samples, namely that homemade samples produce LAB in cocky shape, whereas commercial samples create LAB in rod shape.